we're going to move to the next part of this session, which is actually the introduction to the background document that was sent to you before the weekend. And that is the basis for the discussion on the current state of the art. So I'm going to ask Todd to set up, to put the background document slides, and Lise will be presenting this information. Lise,
thank you.
The webcam is yours.
So hi again. Todd, I will say next for next slide, so it will be easy. So I hope you have all received my email that I have sent before the weekend. It included a document that we have called the background document to the web conference. This document is a compilation of, let's say, the preliminary results that have been drafted by the steering group of experts with which we are working since a few months. So I'm going to quickly go through the methodology that we used, but also to the, let's say, the key findings that were in this document. Regarding the methodology, we decided to identify only recent and peer-reviewed publications, which were studies or in lab or on field, as well as reviews. So it's only publications that were since year 2000. We also decided to restrict the scope of the studies only to wildlife. So no studies that were related to human health, and only a limited number on domestic animals. And also to reduce the scope of the studies to EMR ranges that are from anthropogenic origin, so radiations that are man-made actually. Out of the 147 publications that we first identified, 97 were used for the analysis. So we worked, as I said, with a steering group of experts, which were composed of 4 experts in the field of biology or ecology and 2 technical experts. Next. They produced together what was called an analytical grid. And in this grid, there was the list of all the publications that were used. And for each publication, it was assessed if they were their quality, but also to see to which taxonomic group it was referring, and also which EMR range was studied in the research. And finally, what were the conditions, if this was a lab study or a field study or both. As you can see, the technical experts have made a list of 15 different categories of EMR, and they have, for each publication, assessed which type of EMR were considered in the study. Next. So basically, what was done was to assess the first work was to assess the quality of the publications. On the one hand, the biological experts were assessing the biological and ecological aspects of the study, like which population of species are considered, and and is it an ecological study or is it more at molecular level, these kind of questions. And on the other hand, the technical experts have assessed the technical aspect of the study. So again, which kind of measurements were used for which type of EMR. And they use a rating from 0 to 3 to assess the quality for each study. They also extracted the main findings and identified a few knowledge gaps and research needs related to each study. So next. So I will not go into the detail of the results for each taxonomy group, but just go through the main findings. So the first taxonomy group was on plants, and the work has been done by our expert Alain Vian from the University of Angers. As you can see on the slide, for example, for the biological aspects, it was assessed that more than half of the studies were excellent in terms of assessing the biological aspects of the study. Regarding the technical aspect, also a lot of the studies, more than half of them, were very good. And yeah, the others, as you can see, there were some medium quality and some that were not good, but most of them were technically well set up. Next. Then the key findings were that the most studied taxonomic group were angiosperms, that there were significant changes at cellular and molecular levels, namely metabolic changes and changes in the expressions of the genes. There was also some evidence showing alteration of plant growth and development, both in laboratory and in open field. And yet there were two aspects that remained not answered, which were what were the cellular components that first interfere with EMR, and also what is the reaction of the plants to long-term exposure. Next. If you had the chance to read the background document, you will see that all the statements that have been extracted were assessed with the level of confidence. This means if you have a statement, how well is it established? Is there a consensus amongst the scientific community, and are there many studies that can that are the basis for this statement? Again, I will not go into the detail for each effect, but this is just to show you that the effects have been put on the grid, and that the expert will, for the plants here, will, during his taxonomic session, explain you more in detail what are the effects and how and why he has put them in this place on this model. Next. So regarding the vertebrates, we had around 20 studies. These were assessed by Dr. Pascal Malkemper from the Institute of Molecular Pathology in Vienna. Here, around 40% of them were very good, excellent in terms of the biological and ecological aspects. A third of them were normal, and only a quarter of them were or minimum or not good. And regarding the technical aspect, as you can see on the table, it's more variable, because let's say that half of them were good or very good, and half of them were really or insufficient or really minimal in terms of the technical setup. Next. So for the key findings, the most studied groups were the birds and then the small rodents like mice and rabbits. There was not much agreement on the effects, which this means that almost half of the studies reported effects, but the other half reported no effect. Yet when there were effects were reported, what was found is that there were behavioral changes like disorientation or memory deficits, but also developmental abnormalities, decreased reproduction, increased mortality, and also physiological responses. There were few studies on direct ecological effects. This is the same actually across all the taxonomic groups. This is something that is missing. But if there were still two of them that had some results. One of them, for example, was about the species abundance near effort tower, but it was quite difficult to show that the effects were related to the radiation from other confounders like light pollution. And there was also some evidence on animals avoiding strong radiation sources like radar and mobile phones. Then there were studies on the physiological effects, but unfortunately many of them were lacking theoretical foundation. Yet they were reporting effects on redox reaction and hormone levels, altered nociception, and effects on growth and development. But all in all, regarding the vertebrates, the studies that had the most strong theoretical foundation was regarding the disruption of magnetic orientation of migratory birds by radiofrequency fields. And this would be related to a sensor that is based on a protein called cryptochrome. But there is a more detail also in the background document on this. So next. Again, this is like the few effects that I have just mentioned who are placed in the 4-box model. And again, Pascal Malkemper will give you more detail on this tomorrow. So next slide. Regarding the invertebrates, there were much more studies because at the beginning the request came from BugLife, so the first focus of the scope was on invertebrates, which is why we kept more publications. So these were assessed by Dr. Adam van Bergen from CEH and from Thomas Tjodin from the University of the Aegean. Regarding the biological aspects, you can see that most of them were of normal or medium quality. And regarding the technical aspects there, almost half of them were excellent, but still a third of them were really irrelevant or not sufficient. Next. About the key findings, the most studied organisms were the honeybee, the Drosophila flies, beetles, ants, wild pollinators. Many studies actually have shown that the insect detect and orientate with electrical or magnetic fields. But there were very few ecological studies and no real evidence showing that there is an impact on the biodiversity itself. If we look at the laboratory studies, we can see that most of them focus on physiological or developmental effects, and that they also focus on short-term or acute exposure levels of experimental EMR sources. Unfortunately, no studies on the long-term or chronic exposure level. And they all showed up that the effects are quite complex and variable in direction and effects of size, and even sometimes adverse. And there is some evidence that has been showed on the Drosophila of cell damage or effects on individual development, locomotion, or reproduction. Regarding field studies, there is really a lack of scientifically robust studies. Yet some of them have shown evidence that some field unrealistic proximity to EMR of mobile phones can affect honeybee colony behavior, that exposure to mobile phone antennas are having an effect on, or can have an effect on local wild pollinator abundance, yet but there was no effect reported on reproductive capacity. Next. Again, like for the two other taxonomy groups, you can see that the experts have placed this statement in the 4-box model. And like for the two other groups also, unfortunately, not much of these statements are in on the right up corner. Next. Finally, there are the research gaps and needs that have been identified by them. So for the plant, it would be to use good devices that can improve the quality of exposure, but also to use reliable markers that are inexpensive and that can report changes in the behavior of plants. And and that show information of molecular events that occur after exposure. And also these devices allow a better link with the exposure than the growth studies. These studies should be complemented by growth studies, and the field experiments should then be related to symptoms observed at molecular and biological changes. Next. Regarding the vertebrates, there would be a need to have more research on the definite receptor mechanism used by migratory species to perceive the Earth magnetic field, and also to assess if these receptors are still magnetosensitive in non-migratory species. Again, studies on the effects on long-term exposure and real ecological studies are needed, and also meta-analysis on data sets that take radiofrequency data into account, and also an establishment of fixed radiofrequency measurement stations. Next. Regarding the invertebrates, there is also a need for more ecological studies that would measure the effects of EMR on wild communities, but also on the responses of these communities on the long term in pre and post exposure. These studies should be done over a wide geographical extent and in different natural and anthropogenic systems. There is clearly also a need for interdisciplinary collaboration, and studies should be based on testable hypotheses to avoid ad hoc approaches. And also it's important to have field realistic exposure. And there should be more studies also assessing the chronic effects. And finally, the experiments should examine the potential interplay between the EMR exposure and the foraging ecology and other stressors that affect the nutritional ecology. Next. And finally, the experts have tried to find more cross-cutting and general knowledge gaps and research need. The first one is clearly about the technical setup and quality that would require that there are some standards or parameters that have to be included in the future study to ensure the replication. As said for the 3 taxonomy groups, there should be studies on the chronic and long-term exposure, with realistic exposure settings, mimicking those that are on field. The studies should be hypothesis-driven and based on a theoretical framework that allows making prediction on the outcomes of the experiment. And also there should be a clear distinction between the thermal and non-thermal effects. And finally, the exposure systems that can be used in the field should be really further developed. And this is the end of my presentation. So I thank you for your attention.